Hello, Honors Biology class. I just wanted to give you a quick rundown of the Krebs cycle, which we started in class. I think one of your classes I was able to finish, but also wanted to go over electron transport with you as well. So where we were in class, we have the intermediate steps and the Krebs cycle. And one of the things that I mentioned is that the goal of the Krebs cycle is to really get these high energy electrons. And now remember, I'm going to show you the simplified version of this, and then I'll show you what the actual molecules look like. But I do want you to remember, you don't need to know the actual molecules. So the so what of this? Why do we care about the Krebs cycle? Well, what our goal is we want to capture these high energy electrons with a hydrogen and put them into an electron carrier. Remember, that's going to be our little dump trucks that are carrying the electron and the hydrogen. And of course, we know one of them is NADH. The other one is called FADH2. Now, don't concern yourself about the differences. What you need to remember is that there are molecules that can take up an electron. They can also then give that electron to another process. So in the cycle of all of these molecules, this Krebs cycle, it's also known as the citric acid cycle. But I usually refer to it as the Krebs. You have molecules at each step, and these molecules will have these high energy electrons. It's the energy carriers that are going to come along and grab an electron. And through this process, this is the simplified process, we're going to be able to get 3 NADH, and that's from 3 NAD plus, and they're going to be pulling these electrons and hydrogens with them. In doing so, we're going to be able to break off carbons that have oxygens alongside of it, and those are going to be carbon dioxides, and they're going to leave. We're going to be able to get 1 ATP, we're going to get 1 FADH2, and that is from 1 acetyl-CoA that comes into the cycle. So we have this 2-carbon molecule that joins with a 4-carbon molecule, and so we end up with 6-carbon molecule to start off. That's citric acid, that's the name. So if we start accounting per glucose. Now here's where we need to be careful, because up here at the end of glycolysis we have 2 pyruvate, and that started with 1 glucose. So one thing we've got to be real careful of is this whole diagram is only 1 pyruvate molecule. And if it's 1 pyruvate molecule, that means there's another pyruvate over on this side that's undergoing its own conversion and undergoing its own Krebs cycle, generating its own electron carriers. So everything that happens on the right side happens to the other pyruvate on the left. So let's do a quick accounting. For the conversion step, that's in the rectangle, we get 1 NADH. I'm going to go ahead and circle it. Here's that NADH. However, remember this happens over here too. So the conversion, we get 2 NADH. We also get 1 carbon dioxide, but again it happens with the other pyruvate. So that's 2 carbon dioxide from there. The Krebs cycle will get another 3 NADH. However, there's 3 more that happen with the other pyruvate. So that ends up having 6 NADH. And then by the same principle, 2 times 2, that gives us 4 carbon dioxide. We can get 1 ATP, therefore it's 2 ATP. And then we can get 1 FADH2, but that also then multiplied by 2 FADH2. So suddenly, out of this one cycle, from 2 pyruvates, we're getting a fleet of these electron carriers. And these electron carriers are all holding these high energy electrons that can do something. And that something is what we want to really focus on next. Now, the carbon dioxide is just the wasteful gas. We're going to exhale that out. So let's go ahead and tackle. Oh, before we do that, so this is the actual, these are what the actual molecules look like. But I don't want you to lose sight of that goal. But it is kind of neat to see the fact that you can actually see things disappearing from these molecules. You can watch the carbons disappear. This carbon leaves, and that leaves as carbon dioxide. And as these molecules are manipulated and changed by enzymes, we're capturing those high energy electrons. But again, I'm showing this to you so you can actually see that there are events going on. Sometimes these diagrams are way too simplified. So along comes this electron transport system. And please do add a little diagram. You can hit pause on the video right now. And as you draw this, notice the colors. This right here in orange, this corresponds to the intermembrane space. That's the one downfall with this diagram is it's hard to see what that's pointing to, what that's referring to. Now, there are some things that should look familiar to you. This little lollipop right here, this upside down lollipop, that's our ATP synthase. An ATP synthase, if you recall, in the light-dependent reaction, that's what was allowing us to generate ATP. Well, the same process is happening, but this time we're using the high-energy electrons from glucose. So here are the electron donors. We have NADH, we have FADH2. They're going to drop that electron off at these electron transport molecules. And that electron is high energy. And as that electron gets passed from one molecule to the next, at certain stages, a proton can get pumped. So notice here we have again, we've got our hydrogen ions that are gathering up on this intermembrane space. And we have our high concentration here, and that's going to be really important because that means the protons, the hydrogen ions, can diffuse through that ATP synthase and then generate our ATP. So this should be somewhat familiar to you and almost a bit of a relief, like, okay, we're back on familiar ground. Because at this point, we can really generate all of the ATP that's left generate, because this is coming from the Krebs cycle. So one nice thing to kind of keep in mind, inside the matrix, so here's our matrix. Inside the matrix, this is where our Krebs cycle is happening. So we've got this roundabout of all of these chemical reactions producing NADH. The NADH drops off its electron and then goes back to pick up more electrons. Same thing with FADH2, dropping off its electron and then going back to the Krebs cycle to pick up more. And in that sense, we're going to be able to keep that cycle going. Now, where does this electron go? That electron is going to be really important here because it needs to be dumped somewhere. If it doesn't dump somewhere, notice we've got these molecules holding these electrons and everything backs up. So here's something interesting to think about. We need a little dumping ground of electrons, and it's going to be oxygen. Oxygen's going to be here waiting in the wings to grab a couple of electrons and hydrogens, and we're going to be able to combine them to make water. And so in that regard, we're going to have a place to put our spent electrons. Electrons get dumped in here. They're going to travel. Let me change colors here so you can see it. The electrons are going to travel from molecule to molecule, and then they're going to get dumped into our oxygen to make water. Now, One thing to think about, this is going to be the last thing that we're going to do on Monday, is what happens if we don't have enough oxygen. Because if there's no oxygen to catch that last electron, then this molecule is stuck, and so is this molecule, and so is this molecule, and so is this molecule. And guess what? There's an ADH with nowhere to dump its electron, and then therefore suddenly the Krebs cycle stops. Now, you're probably left with, or you're still wanting to think about all of this, and I completely understand that. What I would like you to do is read over the notes that you have. As soon as you're done with this video, I would like you to read over the notes that you've taken, and try to annotate where possible, add some of the comments, or even think of some of the questions you have for these notes, so that when we do see each other next on Monday, you can come with those questions of, well, wait, where is this coming from, and what's happening here? But I wanted to give you a starting point so that we can hit the ground running a bit when we come back from our weekend. So that's a good start. Like I said, please, as soon as you're done with this video, take a moment, write some questions out, read over your notes, clean up your picture, color where you need to, underline where you need to, use some of those skills that we've been talking about in class to make sure your notes are the best that you can make them. I hope you have a great weekend, and I'll see you on Monday.